Osmosis experiments. Now, one of the most common experiments in our syllabus is cutting cylinders of root vegetables and placing them into distilled water. By distilled water, I mean pure water, and sucrose solutions. Sucrose is a sugar of increasing concentration. This is going to be our independent variable, which we're going to be playing with. Now, cylinders must be weighed before you place them into the solution. We must measure the initial mass and the final mass to then identify the change in mass of the cylinder and identify whether water has moved into or out to investigate osmosis. Okay, they are left in the solution for 20 to 30 minutes, enough time for water to or osmosis to take place, and they must be dried after we are taken out to remove excess liquid and then reweighed. All right, reweighed to compare the final and initial mass. Now, once we do this, there are three possible cases. Okay, if the plant tissue gains mass, this means water moved into the plant tissue from the solution via osmosis. So, if water moved into the plant tissue, the solution is dilute. This means the solution has a high water potential. The plant tissue is concentrated. The plant tissue has a low water potential. So, water moved from a high water potential of the solution, of a dilute solution, to low water potential into the plant, which is concentrated. But if the plant tissue will lose mass, in this case, it is the opposite of scenario 1. The water moved out of the plant tissue into the solution by osmosis. This means that the plant tissue is dilute, it has high water potential, and the water moved out from high water potential into the solution, which is concentrated, of low water potential. But if there was no overall change in mass, this means there was no net movement or osmosis of water, and the concentrations of both the solution and the plant tissue are the same, and it is an isotonic experiment.